And thank you for just the opportunity to be here. I'm very glad to be so early here in Ecuador, joining all of you from all over the world. So, yes, as Victoria said, I'll be talking about a little bit of what I did during my PhD research, with a focus, of course, in extremophiles. And I have to tell you, my passion in extremophiles started, I would say, 12 years ago, when I saw firsthand many of the impacts of mining activity here in Ecuador, where I am from. And I understood then that I didn't know much about the microbes contributing to those impacts, or the microbes related to remediation of those impacts. So I really wanted to know more about them, and the techniques that allow me to do that were metaomics. So here I'll be talking about my research focused on specifically a pit lake that is not in Ecuador, is actually in Spain. Huelva is known as Cueva de la Mora. It's a flooded open pit mine that now is a meromictic and acidic pit lake. It's a very, very well studied lake. It has three biogeochemically well differentiated layers: an upper layer, a chemocline, and a deep layer. All of them are different from each other, as you can see from the chemical data that I'm showing you. And with extreme conditions, especially related to pH, acid pH, and then sulfate, iron, metal, and metalloids like this one, arsenic in this case, are also way higher than common standards. So, microbiologically talking, they are also different from each other. For example, the upper layer hosts green algae and some iron cyclers related to acidobacteria. The chemocline has sulfur and iron cyclers like the ones that I'm showing you there. But the deep layer, before my research was developed, was hosting unknown organisms at that time. This was 2018. And for us, like my group, where I was doing my research at Penn State University, and people in the Geological Survey in Spain, all of us were very interested in knowing the organisms that were living in this deep layer of the lake, especially those related to the sulfide potential biosulfidogenesis, the sulfide production, considering the high sulfate concentration in the lake at its depth, we were interested in knowing if there were organisms able to reduce sulfate to sulfide. And why we were interested? Well, because the sulfide biogenically produced could be actually used as a bioremediation strategy to precipitate out the metals that were also in high concentrations at this depth. So that was one of the main drivers of our research. I wanted to answer these questions. Are there organisms actively involved in specifically the biosulfidogenesis part, the production of sulfide in the deep layer of Cueva de la Mora? And to do that, what we did was basically sample along depth at specific depths. I'm going to talk specifically about the deep layer, sample at 35 meters. We sample water from this depth and then filter them to get these Sterivex filters for RNA and DNA extractions. Then after we extracted the DNA and RNA, we focused on getting metagenomics and metatranscriptomics. From the metagenomics, we were able to assemble metagenome assembled genomes, MAGs, and with the metatranscriptomics, we mapped all of the transcripts we got from the community to the MAGs we already assembled. In this way, we wanted to know which specific MAGs were they referring to, and which activity, potential activity they could have in the deep layer at the time of sampling. So this is part of the results. We had 13 MACs that had at least one gene involved in sulfate reduction, but 5 MACs had the complete pathway with potentially active genes. So here in this plot, in my left, you can see the pathway of sulfate reduction and the enzymes that are related to such pathway. In the right, you see a plot that includes the 13 MACs and also the specific genes that are color-coded according to the plot in my left. So there you can see that the circles in general represent the presence of the genes, but the black dots represent the transcripts that they also have. So in this case, you can see that the 5 MACs that I was referring to have not only the genes, but also the transcripts doing specific sulfate or related specifically to sulfate reduction in the lake. So that was very interesting. That was part of what we wanted to know. But moreover, we wanted to know also if they were somehow related to microorganisms that were already described in literature with this ability of reduce sulfate under acidic conditions, and they were not. So, for example, this was related to the Actinobacteria phylum. We did not find any other organisms related to this one, at least not described before. Then we have this chloroflexi, also unknown as far as we know, and this one that is related to the Nitrospirae phylum that we knew some before. Nitrospirae bacteria able to reduce sulfate, but not under acidic conditions. So of course now one of the focus of the lab in the university where we start doing this is trying to isolate these organisms, but I have to say under laboratory conditions that has actually become a little difficult. We are trying to isolate these organisms in situ with other techniques that might allow us to isolate them, not in the laboratory, but in situ, provided the specific conditions they have in the lake. So, from what we concluded of this data is that sulfide production in the deep layer of the lake, where toxic metals can precipitate as sulfide minerals, is not limited by microbial genetic potential, which is something that we really wanted to have. And well, I'm open to more questions, of course, but thank you so much to, of course, all of my contributors and you that were here listening to me. Thank you.